Good evening friends, you are most welcome to Think High channel. Today we are going to discuss Practicing Anthropology by MAAN IGNOU and our topic is Human Genetics and Society. Let us begin the topic. Contents are introduction part, blood group polymorphism part, hemoglobinopathies, genetic counseling, genetic screening, at the last summary. Learning objectives are: it is expected that after reading Unit 2, you will be able to discuss the importance of blood groups and the procedure in carrying out the ABO and RH-D blood group system. Understand and explain different abnormal hemoglobins and discuss what is genetic counseling and genetic screening and the process of genetic counseling and genetic screening. Introduction part. As you know, physical anthropology is mainly concerned with human variation and evolution. However, within physical anthropology, numerous specialized branches arise such as human growth and development, genetic demography, population genetics, forensic anthropology, human genetics, molecular anthropology, physiological anthropology, applied physical anthropology, etc. But applied physical anthropology is one of the major fields where the knowledge of physical anthropology can be practiced in the society. One such example is anthropometric measurements collected from different populations of the world are used in design equipment, furniture, headgear, garment industry, footwear industry, etc. Nowadays, the understanding of human genetics has turned out to be a central part of our society, and the information of human genetics gained by physical anthropologists can be applied and practiced in society. Hence, this unit focuses on some of the areas of human genetics, namely blood group polymorphism, hemoglobinopathies, genetic counseling, and genetic screening. Physical anthropologists, on gaining knowledge in these areas, are able to practice them in the society. Blood group polymorphism. In blood transfusion, the determination of blood groups is very important. The determination of blood groups is a part of the curriculum for the physical anthropology students. Hence, it is indeed necessary to understand the blood groups. So, we will provide a brief history of the blood groups and the procedure involved in the determination of ABO and RH-D blood groups, the two major blood group systems which are being used in the blood. Carl Landsteiner discovered the ABO blood group system in 1901, and later on more than 20 distinct blood groups systems have been identified. They are MNS, P, RH, Lutheran, Cal, Lewis, Duffy, Kidd, etc. However, till now ABO is the major human blood group system, which has four major blood groups: A, B, AB and O. All people can be placed into one of these four: A, B, AB and O blood groups based on the presence or absence of two antigens and two antibodies. The antigens designated as A and B and the antibodies anti-A and anti-B. The antigens are present on the red blood cells while the antibodies are found in the plasma. The following table shows the antigens and the corresponding antibodies present in the blood group. AB blood group is considered as the universal recipient and O blood group as the universal donor. Blood groups antigen antibody can donate blood group to and can receive blood from. तो ब्लड ग्रुप ए में हम देखेंगे एंटीजन ए होगा, एंटीबॉडी एंटी बी होगी। डोनेट किसको किया जा सकता है? ए को और ए बी को, और रिसीव किससे किया जा सकता है? ए और ओ से। ब्लड ग्रुप बी में हम देखेंगे एंटीजन कौन सा होगा? इसका बी होगा। एंटीबॉडी एंटी ए होगा। इसको किसको हम डोनेट कर सकते हैं? बी और ए बी को। और रिसीव किससे कर सकते हैं? ले किससे सकते हैं? बी और ओ से। ए बी ब्लड ग्रुप की बात करते हैं, एंटीजन ए और बी होंगे, एंटीबॉडी कोई नहीं होगा इसका। इसको किसको डोनेट कर सकते हैं? ए बी को कर सकते हैं। रिसीव किससे कर सकते हैं? ए बी और ओ से कर सकते हैं। ब्लड ग्रुप ओ की बात करते हैं, एंटीजन होता ही नहीं इसमें, एंटीबॉडी एंटी ए और एंटी बी होगी। डोनेट किसको किया जा सकता है? ए बी और ओ को। रिसीव किससे कर सकता है? केवल ओ से। प्रोसीजर फॉर आइडेंटिफिकेशन ABO blood group. Two methods are in practice. One is porcelain tile slide method and other one is tube method. Tile or slide method. Place 1 drop of anti-A serum in one cavity of the slide or tile and same quantity of anti-B serum in another cavity. Add 1 drop of 10% cell suspension already prepared from the collected blood sample in normal saline to each cavity and wait for 2 to 3 minutes and observe the agglutination reaction. If agglutination is observed in the cavity where anti-A serum was added, it indicates A blood group. If the reaction occurs in the cavity where anti-B is added, it indicates B blood group. If agglutination is observed in both the cavities, the blood group is AB group, while lack of agglutination in both the cavities indicates O group. Tube method. Take 2 small test tubes and add 1 drop of anti-A serum into one tube and 1 drop of anti-B serum into another tube. Add 1 drop of 3 to 5% cell suspension already prepared from the collected blood sample to each tube. Mix the contents gently and allow them at room temperature for few minutes. Then observe for agglutination reaction. Interpret the results as stated for the tile or slide method. RHD blood group. This is another major blood group system which is also used in transfusion medicine. The RH blood group system was discovered in 1940 by Karl Landsteiner and Wiener. This blood group system is based on the presence or absence of RH antigen, frequently called the RH factor. The term positive or negative refers to either the presence or absence of RHD antigen. While blood grouping, if the blood agglutinates in presence of anti-RHD antibody, it indicates as positive, and if the blood does not agglutinate, it indicates as negative. Procedure for identification of RHD blood group. Place 1 drop of anti-RHD serum into a microtube and add 1 drop of 5% cell suspension already prepared from the collected blood sample. Keep a contents at 37 degrees centigrade for minutes and then observe for agglutination reaction. If agglutination occurs in anti-RHD serum, the RH factor is positive, and if it does not, the RH factor is negative. Hemoglobinopathies. This is another important area of human genetics. The information gained in this area may be practiced in the society by physical anthropologists. Before going into details of hemoglobinopathies, let us have a brief look on the normal hemoglobin. Hemoglobin is a tetramer that consists of two alike and two alike globin subunits. Hemoglobin is an air-containing protein of the red blood cells. There are 4 kinds of hemoglobin: the embryonic hemoglobin, the fetal hemoglobin HbF, adult hemoglobin HbA, and hemoglobin A2 HbA2. Under normal condition, the red cells of an adult man contains approximately 98% HbA hemoglobin adult, traces of HbF hemoglobin fetal, and 2% of HbA2 hemoglobin A2. On the other hand, hemoglobinopathies are the inherited disorder of blood. Following is the brief account of hemoglobinopathies. The inherited disorder of hemoglobin comprise hemoglobinopathy and thalassemias. Hemoglobinopathies comprises a major bulk of non-communicable genetic diseases and one of the major public health problems in many countries including India. They are most widespread in ethnic populations from Africa and Mediterranean basin and Southeast Asia. The vulnerable segments of the society like infants and children, adolescent girls, pregnant women, etc. are severely affected by these abnormalities and so cause a high degree of morbidity, moderate to severe hemolytic anemia. The most frequent mutation of hemoglobin which affect the beta globin gene and result in a hemoglobinopathies are sickle hemoglobin HbS, hemoglobin C HbC, hemoglobin D HbD, hemoglobin E HbE, hemoglobin O Arab HbO Arab, but in India HbS, HbE and HbD are widely prevalent. But thalassemias are hereditary disorder of hemoglobin synthesis characterized by reduced rate of production of normal hemoglobin due to absence or decrease in the synthesis of the amount of the globin chain produced. It can be divided into alpha thalassemias, alpha thal, and beta thalassemias, beta thal. Beta thalassemias is again of two types, thalassemia major and thalassemia minor. Thalassemia major causes severe hemolytic anemia while the thalassemia minor causes mild anemia. The diagnostic method for hemoglobinopathies are discussed briefly. Diagnostic methods. The abnormal hemoglobins or hemoglobinopathies can be identified by various laboratory methods which include hematological examination, electrophoretic techniques, estimation of fetal hemoglobin HbF, determination of hemoglobin A2, HbA2, sickle cell examination, high pressure liquid chromatography, HPLC, etc. Hematological examination consists of determination of total hemoglobin concentration gram per 100 ml, number of red blood cells 10 to the power 6 per millimeter cube, and the percentage of packed cell volume PCV, hematocrit, naked eye single tube red cell osmotic fragility test, NESTROFT, NESTROFT. Then calculation of different red cell indices. Then calculation of different red cell indices such as mean corpuscular volume MCV in micron cube, mean corpuscular hemoglobin MCH in microgram, and mean corpuscular hemoglobin concentration MCHC in percent. For calculation of the red cell indices, the following formula is used. MCV is equal to hematocrit percentage into 10 upon RBC count billions per millimeter cube blood. MCH is equal to hemoglobin gram per 100 ml upon hematocrit percentage into 100. MCSC hemoglobin in gram into 100 upon hematocrit percentage. However, these red cells indices can also be obtained through standard electronic cell counters. To diagnose hemoglobin abnormalities, electrophoretic technique is an easy and convenient method. This can be performed either on paper, in starch gel, on cellulose acetate, in agarose gel, and acrylamide gel, The fetal hemoglobin can be determined with the help of alkali denaturation, blood slide, and electrophoretic methods. But hemoglobin A2 can be determined by using electrophoretic technique. Different methods are available to diagnose sickle cell anemia. Bhasin and Chahal 1996 they said about that, and they are Scriver and Walk method, bisulfite method, and Cycladex method. But most common method to diagnose sickle anemia is bisulfite method. But most common method to diagnose sickle anemia is basalphide method. But recently high performance liquid chromatography technique has been used for the diagnosis of hemoglobinopathies and thalassemias. Their advantages over other methods include sensitivity, resolution and simplicity as well as speed. Thank you very much.